Hello, welcome back to our last class in level 9, where we're going to bring all those lessons together and address the question, does doxycycline affect attack or epigenetic sites in the genome from going from open chromatin to closed or closed chromatin to open? And we've, through the last series of 5 classes, figured out that it doesn't. So we're going to formalize that and make a nice document that combines all of our analyses, and that's in lessons 09, level 9, 06, how to knit all this together and put it together. And I'm going to go to my our studio environment, and let's do this. I loaded the thing that's going to take a minute, but these are all the packages you would need to do a nice, beautiful knit, and I'll show you just sort of as a preview of what this is going to look like with nice figures, code, results, and at the end, some nice, well, not that part, but some nice conclusions and plots. So I'm going to go through this really fast. This all knits. Like, you can actually just hit run this whole document, hit this knit button here, and it'll work. Assuming you have your peak path correct to your peak files for the ATACseq and your RNA-seq analyses as we've been loading them throughout the previous classes. Okay, so let's set our peak path. This is where all my broad peak files are from ATACseq. We're going to do this FL, make that so that we can, oh no, I ran the thing that I ran earlier so that it wouldn't take as long. Well, we're going to upload all of our peak files that are in there. Those are the 12 files. And this is more for the knitting part, is we're going to do print a sentence and then the number of peaks per sample here. And then when we look at the knitted document, we'll see that printout here like this. Here's the result, the roughly how many peaks there are per sample. Okay, so to find common peaks, we can use our find common peaks function. There's some warnings, so I put the suppress warnings here so that when I knit this into a nice web document, those warnings won't show up. Okay, so now we have the peaks that are common in all of our samples. Here's some markdown to make a nice figure that will show up with a title when it's knitted here. So we'll see this, and we can say this figure shows blah, 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 and there's this many. Here's what a common peaks looks. So here's this box is what you've seen in IGV, is where a peak interval is, and here's a peak that was called in all the samples. It looks pretty good compared to regions that were not called, and it's on this gene here. That's just one example. I had to make a picture of this, put it in my results folder, and then index it here is the way of getting it to show up as a figure, is exclamation mark, square brackets, etc. Okay, I'm not going to go over those for each one, but we can make our peak files of our non-dox samples, which are the 0 time points, and our dox. We can find the common peaks that are in all those 0 time points, say how many there are. This is the same code we just went over. That now we're going to make another thing called dox samples, which is everything that's not 0. And we can say how many peaks come out of that. And now we're going to use our find common peaks to find overlaps between our non-dox common peaks and our dox common peaks. So how many of those overlap, and then we can see that how many overlap, meaning there's 16,000 that don't overlap, and we can now use find my peaks to find the unique peaks that are in the 0 time point and not in the other dox time points. And we've done all this. This is just putting it together into one document. And so now we can say the peaks that are unique to dox is doing find my peaks the other way. It tells us that. Now here's some examples of those peaks. They don't look too convincing that they were in dox and not in the other one. Here's a bunch of results and summaries. I won't spell all these out. And then we need to load in our genome annotation, which is going to take a minute. But when you knit a document, you want everything to be able to run from scratch. And so that's why we're not loading all those files. We could have, but it's just better to put all the code together into one document. So we'll come back in just a second. when this loads. Okay, it finished, and so now we have all the genome annotations uploaded as G-ranges, as we've done in previous classes, and now we're just going to make an object of just the genes in mouse, and then we'll make promoters around all those genes that are 2,000 bases up and down using the promoters function. Do the same thing for mRNA genes, mRNA promoters, lincRNA genes, and we have saved all that stuff and we could have loaded it, but I just think this way it's more reproducible that you see each step as it goes. Okay, so now we're going to do some of these find common peak overlaps again, and or other indexing here. And so this is basically the same thing we did last time. We just have some suppressed warnings in there, and there's this is how many non-docs unique peaks overlapped with gene promoters. So how many, there's about 2,178. Again, we're just making results that we have already made before, and it's printing them out as we go to make it into one nice document. Now here we did load our RNA-seq data. So we got our res-df, which has the genes that were significant by RNA-seq. And we can say sig RNA-seq attack peak overlaps in non-docs by just filtering the gene IDs between those two. Again, printing out the result. I'm going really fast through this because we've already done all these, but it's good to go pause the video and go through and look at what is happening in these indexings. Again, we've done them all before, and that's going to just print out the results here. Okay, and here are all of our results. And now here we begin the DE-seq thing, which we just did in the last class, where we're going to load our count file, where each peak is now like a gene for DE-seq, where each interval, and we're going to do DE-seq, make the sample sheet, and clean that up. Again, that was done in the last class, 05. All the same things, and we're running ATACseq based on condition here, which is just non-docs versus docs. We saw that no genes changed in all the time point comparisons, which would have been an easier way to find genes, but when we do DE-seq for, so we're just going to do it for non-docs and docs, because the answer is no peaks change significantly in either of those things. And all these warnings that are coming out are being suppressed in this suppressed warning, where we're running DE-seq on the condition of our sample sheet and suppressing the warnings that are coming out as we go here. And then we'll make that data frame that has the DE-seq p-values, fold change, et cetera, for each peak. So you have a really handy table, and this is why DE-seq is taking longer, is there's like 174,000 peaks in this counts file. So those are all the peaks that occurred in any sample. Okay, so now we can grab our results with the results function, make that into a nice table, selecting the columns we wanted again. And now this is just plotting all those plots we did in the last class, that there are no p-values that change, and then these conclusions are written out here. And so voila, you can hit, this whole document runs, as you can see, and it will work. So this is what you will get, and so you can open this in a web browser and send it to somebody, and they'll look at all your research and say, oh, okay, I'm looking at peaks, now I'm going to do DE-seq, and here's the fold changes and p-values, I don't see anything. This part you might want to delete because it's just more instructions on how to knit. But congratulations, this is an impressive document you have here, and there's a lot more we can do, but you now have all the fundamental skills to not only do RNA-seq, find genes that are significantly different across time series, do the same thing for ATACseq peaks and overlap them, and then also overlap them with genome features of mRNAs and lincRNAs. So I know it seems like a lot, but you have all the documentation, you know where all the code is, and in level 10, it's choose your own adventure, where we can put this together and try and write a paper, and I think there's some really interesting results we've already found from class. So there's a lot of potential here, and in the level 10, the final one will only be one video, and I'll just outline some things you could think about doing for your analyses, and some more on where files can be found for people taking this publicly. All right. Thanks very much. Congratulations. You've done it all. You can do RNA-seq and ATACseq and overlap them, and you answered a really interesting question about whether DOX affects stem cell gene expression, and it does. It affects hundreds of genes, but it does not affect the chromatin state as measured by ATACseq. So you are now people that know that. Congratulations. And we'll see you on level 10, where maybe you could make a figure and contribute to a citizen science project to make this into a primary publication. Thanks very much.